Okay, welcome everybody. Happy Monday. So we left off about halfway through this chapter. We left off with some good ones, I feel like. Schistosomes are very disturbing to me. I feel, when I think about all sorts of different types of infectious disease, parasites specifically, and we're going to shift over and talk about some fungi in a minute as well. There are boatloads of these that I step back and say, I am so happy that I live in Idaho. We don't think about these things here. We only think about them if you've traveled someplace else. That's typically what we think about. And I'm pretty happy for that. There are definitely some potentials, and fungi, it's getting a little more on our radar as we are looking at some of these things, potentially expanding their geographic locations, thanks to some changes and shifts in climate that make it a little bit better for some of these things maybe to be able to survive. That stuff, it just kind of makes sense that if the habitat becomes a little better, they may show up, all right? The schistosomes, we don't see the ones that cause serious, potentially fatal infections in people. We don't see these things here, but we do see some of them that, cousins, I guess, they're still schistosomes. There are lots of parasites that can infect all sorts of different things. And sometimes they jump ship and infect a different new type of host. And sometimes they get into that host, but they can't really do their job. They can't complete the migrations like they need. And they stay relatively localized, if you will. All right. Swimmer's itch is something that happens here. We definitely have some opportunities, and in the heat of the summer, people like going swimming in all sorts of different lakes or ponds. And if it just so happens that there is the right type of snail, there are potentials for us to maybe get infected. But in these circumstances, we're not the right host. They really would like to be infecting birds, and then the life cycle would be completed. And in us, they can't. And so they stay localized in the skin. Our body mounts a defensive response against them, and we kill them off. All right. They don't progress. All right. But you definitely feel uncomfortable. All right. And in southeastern Idaho, there are definitely areas where you can contract swimmer's itch. All right. Some of you maybe had it. All right. So a relative of those that actually cause human disease. There are three big ones that we've got on this image, but technically there's a couple more that can cause some issues as well. We oftentimes think about areas like nice warm tropical locations. Africa, pretty good for some of these schistosomes, but we can see some in Central and South America as some possibilities as well. All right. They like fresh water. That's a potential. All right. So keeping out of the water can keep you free and clear of some of these. Sometimes you just want to get in there, right? So the life cycles of these schistosomes, they are a type of fluke, so a flatworm, but they're a little bit different. They're a little odd compared to the others. And I mentioned them, the flukes and the tapeworms as having hermaphrodism. So both sex organs in a single worm. In these, they're separate, and they are truly an exception when we think about the flatworms as far as how they reproduce, right? These different, right? And in turn, upon infection, love is in the air. They find one another, they pair up and mate, and that's what we've got going on right here. The male and female intertwined, they tangle up. The male has the schistosome means split body. It's got a big groove, we call it a gynecophoral groove. It's a split opening in this worm, and the female wriggles in between there. He clasps around her, and they are paired up and locked in love for the rest of their lives. All right. Basically, constantly copulating, all right, and pumping out eggs. I'm like, somebody's going to need a drink of water, right? That's an issue, all right? They are continuously reproducing and producing these eggs, which really are what's causing the issues in us, not the adult worm, right? They're a bit of a problem, but really it's the eggs that are more of the issue, all right, that can lend to some ill effects and potentially, in the case of hematobium here, maybe lending to bladder cancer. They can live in us for decades. And so the potential for ill effects get compounded the longer you're living with these guys, all right? So the life cycles are super complex. It involves us or maybe some others. With some of these that infect humans, we can see some non-human primates serving as reservoirs, but typically we think of ourselves as definitive hosts, right? So, and we are the ones where reproduction is occurring and they're pumping out the eggs, all right? So dependent upon the circumstances, all right, we either, and I'm gonna skip ahead a little bit because that's right where I'm talking. The worms find one another, they pair up, they start mating, and the first two, Massinae and Japonicum, they make their way down into the mesenteric venules of the bowels or the rectum. They lay their eggs and in turn shed in the feces. Hematobium, it goes to the venous plexus of the bladder. The eggs get released into the bladder and in turn voided out in the urine, all right? We can't see it very well right here. I guess we can right here. Here, this is a hematobium, schistosoma hematobium. This one that would be in the bladder, voided in the urine, and they have potentially have little spikes or spines on the egg surface. That in turn can mount some ill effects and causes damage in the bladder, lending to calcification or alteration in the squamous cells that lend to different problems for sure, all right? Maybe lending to cancers, all right? Mansoni has one that's a terminal spine, and Japonicum is just a little rounded bleb. It's not really a spike, all right? So you kind of differentiate based off of the eggs. You also could differentiate based off of where it's coming from, your urine or feces, that would clue you into some of those details. So the eggs are released one way or another out of our body. And in these instances, the hopes for this parasite would be that your business was done in the water or it's able to contaminate the water. These eggs get into the waterways and in turn, they hatch. There are multiple steps within here. What hatches out of this egg is called a myristidium. It's a little ciliated organism, which looks like a protist, but it's not. It's just the juvenile stage of the worm, all right? So this little ciliated myristidium swims around hoping to find a snail, and it actually can sense or seek out the scent of snail mucus, and it swims very rapidly toward those snails, making contact with the snail and boring into them, augering in, and as it twists into the snail, it sheds its skin, its ciliated epidermis, and in turn becomes the next stage in the life cycle. We call it a sporocyst. And there are all sorts of different potentials with these. They may replicate and get another round of those, or we give rise to the next step. Basically, it's really kind of like a structure that houses the next step, all right? Which is really pretty crazy to begin with, all right? So those sporocysts, there's rounds of divisions, asexual divisions inside of them, producing things that we call rediae. And then those rediae produce the next step called cercaria. The cercaria are little flagellated forms that emerge out of the snail, bore out of the snail, and they get into the fresh water swimming around. Now, their hopes are to come in contact with us, right? When we're talking these guys. And as our skin is in the water, they make contact, they penetrate in very rapidly, and then they migrate, all right? If we're talking the swimmer's itch, they penetrate into our skin, and that's as far as they go. They stay localized, but not these, all right? So they migrate through the circulation, and as I said, pairing up, mating, releasing the eggs, cycle complete, all right? Lots of really kind of impressive stuff and, you know, disease manifestations. We're not going into all of that stuff right now, but you can have some pretty serious organ involvement, liver and spleen swelling up really tremendously with some of them. The Mansoni and japonicum for sure. Hematobium, not so much with that. So pretty impressive, I think. All right. The next ones we want to look at briefly, I guess I would like to look at them for the whole hour, are some tapeworms. There are lots of different potentials when we think about tapeworms. Two of the main ones really kind of on our radar are beef and pork tapeworms, right? And Taenia saginata and Solium are the species of those respectively. There are others, like some that we may potentially get through the consumption of fish that is raw or poorly cooked, that may lend to other tapeworm infections, right? Kind of just depends on the circumstances. And it also potentially depends on how that fish was dealt with, all right? Fresh caught, never processed in any sort of way, never frozen. You definitely have a greater likelihood of contracting some tapeworms through fish that way, all right? Meat, like beef or pork, similar idea. If it's not cooked thoroughly, the cysts, larval stage, if you will, those cysts are in the musculature of that animal. We ingest that stuff, and in turn, they hatch out and we get the adult stage of the worm, all right? That's the issue with it. So, dependent on what it is you're consuming, this is some of those cysts that are in the musculature that we consume, and then ultimately within the intestines, we get the adult stage. And these can be truly a pretty impressive size, meters, pushing maybe 20 meters in length. In the United States, they typically don't quite, the beef tapeworm's one of the bigger ones, not quite that big, maybe 5 to 10. But if we look elsewhere, they can be a little bit bigger. And I don't have really an answer for why that is, but that's just the way it is, right? There are potentials in the US to contract some of this stuff, but it is relatively low, all right? So keeping that in the back of your mind, all right? Not that I would recommend eating, you know, a rare pork chop, right? But a rare steak, maybe that's delicious. Why wouldn't a pork chop be delicious, right? Because in the back of our mind, we're like, you're gonna get worms, right? Probably could get worms from all of them, right? So here we can see just a little blurb here with a cross section of the proglottids, the segments, all right? And as I described, each one of these segments has reproductive structures. The little proglottid gets full of eggs, and then they in turn either rupture and you defecate out those eggs, or you break off parts of the tail of that worm, and you get a segment of chains of proglottids that you release. That might be your first instance that you got something going on, all right? If it's simply releasing eggs, you may not know it, all right? So that's the idea. With the pork tapeworm, it is a little bit different, all right? And technically the pork roundworm also a little bit different. And we've got an opportunity with both of those where we could serve as intermediary hosts and the definitive host. And it comes right down to like hygiene issues. If you're not washing your hands properly after you've used the bathroom, you've got some potentials if you've got these worms, right? Or if the person preparing your food has these worms and in turn doesn't wash their hands, that sets the stage for you to ingest the egg. And then we serve like the pig. The egg hatches out in our stomach, bores through into the musculature, and it insists. There it lays in wait, hoping to be consumed by something, right? For us, probably a dead end, I would think, right? So with pig tapeworms and roundworms, we could be both definitive hosts harboring the worm in our intestine and intermediary hosts where we harbor the juvenile or larval stage encysted in our musculature. We don't see that with other worms, right? And there are, numerous things that, pigs and people have some similarities genetically. Potentials for stuff like heart valves to be taken out of a pig and put into a person with very little issues of rejection are huge. And that sets the stage thinking about all sorts of other infectious diseases. And for them to jump between, it kind of makes sense, right? So all sorts of possibilities with that. And, we show similar stuff with them both together because the life cycles are truly very similar. It just depends on the type of animal that you could potentially be consuming, all right? When we think about some different countries, maybe some different religions, that sets the stage for some potentials to maybe not have these on their radar. If you're not eating beef or you're not eating pork, well, you typically don't think about ever having those issues, right? But if you're not eating the one, you're probably eating the other, right? And so that opens up the potential for worms. Everybody's got those opportunities, right? Like, but I'm a vegetarian, right? You can get worms from eating other stuff too, right? Vegetarians aren't protected, right? There's potentials. Liver flukes are a good one for some aquatic vegetation where you could ingest some of that stuff and you get that. Some of the fasciola. We showed an image of that last time, I believe. This image, I think this is super impressive. There are lots of diseases that, have plagued us for forever. And lots of things that we're like, man, couldn't we do something about that? Couldn't we prevent it? Couldn't we cure this so we never even have it on our radar at all? Oftentimes, that's not the case, all right? But with our guinea worm, we are super close to saying we have eradicated this. It's almost not a human concern anymore, all right? Almost. There, in the past handful of years, there have only been literally a handful of people that have been infected with the guinea worm, all right? So maybe, all right? With that said, there's actually some issues because Fido right here, it's a problem, all right? We may be free and clear, but dogs and maybe some other stuff, but dogs definitely seem as if now they're picking up where we are saying we're done with you, right? The worm still wants to infect something, right? So the idea, typically, we get infected through drinking contaminated water, but there are some potentials where we may eat a transport host. We call that a parentenic host. So something that ate this little invertebrate, this copepod, and then we eat it raw or poorly cooked again, and then it gets into us. But typically, it's the water, all right? So the parasite is inside of this copepod, this little aquatic invertebrate, all right? And it gets accidentally ingested, drank, all right? And then once that happens, worms are released. Guinea worms are a type of roundworm, a nematode, and we can see females that are huge compared to the little males over here, all right? Females can be upwards of 1 meter long when they are fully grown, and the males, we're talking maybe a centimeter, handful of millimeters, but probably a centimeter, all right? Not very big, all right? The life cycle of this, once we have drank down that copepod, before we get to the point where we're seeing this individual's ankle twisting a worm out, it's about a year after you drink that copepod before she's ready to emerge and start pumping out eggs, all right? It takes a super long time, all right? So after we've ingested, they pair up, they mate, the male dies shortly thereafter, she's producing these eggs, she starts migrating down toward your appendages, your foot is where she really would like to be, all right? As the eggs are ready to be released, a big ulcer starts to form, typically again in the foot or lower ankle right here. That's the Achilles tendon right there. That sucker's gotta hurt, right? And in turn, she pokes out and starts releasing the larval forms, some juveniles, into the water. Those in turn attract the attention of these copepods that think yummy food. They feed on them, and in turn, we drink them down. Through our understanding of these life cycles and recognizing these issues, areas where these are endemic, we have gone to great lengths to ensure that people are getting some fresh water or have some filtration potentials to ensure that the copepods aren't being consumed. And that's truly driven a wedge in the life cycle of these guys. But like I said, dogs, they've kind of picked up the slack. And if you've got dogs and you take them anywhere where there's a bit of water, they're going to drink that stuff, right? We just have to, right? Need a little drink, right? Just to see what's up, right? And in turn, that's where some of those potentials remain. So it's not like we completely kill them off, right? They're still there. It's just a matter of maybe we're not as likely to get it. So here we could see a matchstick where we're twisting it out, and it's still a mechanism to deal with these worms. Removing them, once they emerge, you basically try to slowly, kind of methodically, turn them out of your body. And to twist that meter-long worm out takes probably a couple of weeks of slowly twisting. You can't just pull it out, right? Because it's likely you're going to break it and cause other issues with the dead worm and our body mounting a response against it like that. So it's slowly removed from the skin, which to me seems disturbing in and of itself. All right. Pretty crazy. All right. So shifting away from the parasites, from the protists and the helminths, we want to think about some other eukaryotes, some yeasts, or molds. These are molds. Here's yeasts, although they look sort of similar. There are all sorts of potentials when we think about fungi to cause problems, from yeast infections to ringworm or athlete's foot to some definitely more serious pulmonary infections that we might think about from some of the fungi that we call dimorphic fungi. Those that may live in the environment as a mold, releasing spores to be inhaled and converting into yeast cells that replicate pretty rapidly inside of our bodies, right? And those are some that are kind of, in the headlines talking about potentials for moving maybe into our habitats right up here, thinking about some potentials with dimorphic fungi setting the stage for problems, all right? There are ways to look at them and evaluate. There's also differences in their life cycles, from sexual to asexual forms of reproduction and the types of spores they produce. I'm not going to get super bogged down with all of that stuff, but I will show you a couple of pictures for some of the variations. But when it comes to the molds, the individual threads or strands of that mold, we call hyphae. And the whole collection of it, we would call mycelium. Mycelium is just a boatload of hyphae, a bunch of threads, right? And if we were to look at this hyphae under the microscope, it might look like little divisions, little cell walls in between each of these that we call septate hyphae. Or it may look like one big continuum, one big strand that's multinucleated, if you will, because there aren't any divisions. And we call these non-septate or cynocytic, for just a continuum, all right? Each of these dots is the nucleus, all right? And then these yeast cells, there are some potentials with them to produce some substances that look like hyphae. It's not true hyphae, all right? It's just kind of an elongation of their cell that looks sort of thread-like, if you will. And that may lend to some potentials for virulence, for enhancing their ability to take hold and establish some disease, if they can do that. So this first one is a type of dimorphic fungus, Histoplasma capsulatum. It is one that we really think about these guys, Mississippi River Valley and east is more where histoplasma is located. We might be pushing a little bit west of the Mississippi, but not too much, all right, when we think about it. Although now, definitely some opportunities for some more expansion, all right? But typically we think of some geographic locations. That's really kind of it for these guys. There's another fungus called Blastomyces dermatitidis that has kind of a similar overlap in geographic range. Mississippi River Valley east from southern Canada all the way south, all right? Those are potentials. However, Blastomyces has been isolated in some other kind of novel habitats. It's been found in Colorado. It's potentially got really a pretty tremendous geographic range, and we've just yet to really understand that, all right? The other dimorphic fungus that's a big one in the US is called Coccidioides imbitus. Valley fever is that one. And it's found in the southwestern parts of the United States. The San Joaquin Valley in California is the valley fever, and it is pretty endemic to find those fungi growing in the soils around there, all right? So those are the three big ones, and we'll see a little bit more of that, and we'll definitely see more of it later. But as we're talking a little of that, we can't just say histo and leave it at that, right? Those are the main ones, those three that we really think about in the US, all right? There are a few others as we move abroad, all right? So in this case, histoplasma, like some situations in its environment that are more kind of nutrient dense, so high nitrogen, bat droppings are a good one, but it also could be some bird droppings. Bats are better, a little bit better for it, all right? So here we are up in an attic with some bats. You hope you don't have bats in your attic, right? And this is the guano, the bat excrement, that in turn is pretty good nutrients for this fungus, and it's thriving in there. This is a microscopic view of the hyphae and the conidia. These are the reproductive structures where the spores are housed within this conidia. They get released out, airborne, and in turn, we've got an opportunity to inhale that. All of these dimorphic fungi that I've mentioned start off in the lungs as a pulmonary infection. Depending upon maybe the health of the individual, you might recognize it, or maybe you don't at all, and it's asymptomatic, all right? Just kind of depends, all right? So within the lungs, they start to grow and proliferate as yeast cells, and then the potential for these fungi to disseminate and to go elsewhere in the body is huge. And we call these systematic infections. It's infecting more than just your lungs, all right? So moving into the circulatory system or the lymphatic, spreading throughout the body. Oftentimes with these, the first sorts of outward manifestations, not pulmonary stuff, would be some skin lesions that start showing up on the body, making you wonder really what's happening, all right? If you don't catch it at the pulmonary stage. By the time it's disseminated, that makes it considerably more difficult to treat, all right? So with the fungi, there are all sorts of different potentials. And we think about some, like grouping them up based off of how they reproduce. Sexual cycles and asexual cycles are oftentimes seen in these fungi. We kind of assume that they probably have both, but there are some where we may only recognize an asexual life cycle. It may not really be accurate. It might just be that that's all we've been able to recognize at this point. And so there's multiple ways that we group them based off of how they can reproduce, all right? Zygomycetes have sexual and asexual life cycles. And with this, we're not talking male and female, but it's kind of like that. We call them opposite mating types, positive and negative mating types, all right? And when two hyphae of opposite mating types come pretty close together, the potential for them to form a union to reproduce is there, all right? That's down below. Oftentimes, they just reproduce asexually, generating these spores through a process of mitosis, and they continue to grow on as more strands. But given the opportunity for opposite mating types to be close together, they can fuse, forming this little fusion in here, gametogamy, as they do this. We call it a dikaryon or karyogamy, where they basically fuse, the two cells fuse, and the fungus is haploid, and then it becomes diploid, if you will, during this fusion. But really, it's two nuclei, right, to the dikaryon. Not the same as we think about sexual reproduction, but pretty much the same, all right? So we say this zygote is diploid here, and then we go through our meiosis generating the sporangium, the sac that's going to harbor the spores that are back to the haploid state, right? So it has an opportunity to reproduce in different ways, asexually, just kind of cloning itself, and then a recombination, if you will. There is some potential for some genetic exchange with these opposite mating types, giving some alterations a little bit within the genome, if you will. Lots of them go through processes that are similar to this, where opposite types attract, right? So here we can see some bread mold, and all of it, we would say mycelium, the individual threads that we've got in the microscopic image, the hyphae, all right? And then the sporangia, the reproductive structures, that's often kind of the pigments that we can see in some of these molds, blackish or greenish looking stuff. Oftentimes that stuff is where the spores are housed, all right? And that's what we're seeing here, all right? So here we've got mucor, and I think that's this first bit. Mucor fungus found indoors, potentially could be found on some bread. Rhizopus is also a good one for bread, all right? And these two, mucor and rhizopus, really pretty common to think about as far as fungi goes. My gut says most of us have probably seen some moldy bread, right? Probably, and it's likely some rhizopus that we've seen, all right? Some of us potentially maybe ingested some of that, not knowing, right? You make yourself a sandwich and then you're just about done eating and you're like, holy crap, this bread was moldy, right? Gross. Or maybe you're like, is that why it tasted so good, right? And then you just want to eat moldy bread, right? I was talking about some of this was years back, And a student raised their hand and said it was their roommate. It's like, oh, I've got a friend. And as soon as they say that, they're talking about themselves, right? But they just don't want you to know, right? Oh, I've got a roommate that likes to wait till this bread's moldy and then they make their sandwiches, right? And I'm like, well, that's weird. I wonder why. And the person says, oh, they say, right? They say that it tastes better that way. And I'm like, oh, do they? Right? Gross. All right. We potentially consume some moldy stuff. If you're eating, like some blue cheese or tempeh or something like that, there's molds that's used in the production of that, but it's not this, right? There's all sorts of different things that maybe we could consume, and there's other stuff that you probably don't want to, right? So these relatively common, all right? But here's something that's a little bit disturbing for you. You'll never think of moldy bread the same way, all right? If you happen to be severely immunocompromised, all right? If you've got some serious issues, all right? And maybe your immune response is just non-existent or very depleted. Maybe we've progressed from HIV to AIDS and we've got no mechanism to deal with anything, all right? You could open up a bag of bread and just inhaling a few of these spores in about 2 days, you're a goner, all right? As they get into your body, make their way into your brain and kill you. I love it. That stuff is nuts, right? But like I said, we've all probably seen that stuff. You've all probably, opened that bag, pulled the bread out. You're like, oh, moldy, and you throw it away, right? You've probably inhaled a few of those spores. Our immune responses in a healthy individual, drill those guys. You can't let them persist, and we don't have any issues with it, all right? But if your body can't mount an adequate response, this thing that's sort of all over the place and we think of as not that big of a deal truly could be the death of you. That it's terrifying to think about some of those things, right? So I've got 2 kids. When my wife was pregnant with the first, I saw some changes. And, you hear about all sorts of things like, oh, they all of a sudden they want to eat this or they want to eat that or whatever it might be, right? So I had some thoughts like, wow, maybe she will want to eat a nice steak, right? Because she's vegetarian, right? I'm like, sweet burgers, right? I mean, I'm eating that stuff anyways, right? But I'm like, maybe she will decide, right? This stuff is delicious, right? No, she had no shifts in appetite, but her sense of smell was severely heightened, I would say. She could smell stuff that I'm pretty sure she's making that stuff up, right? I open up some bread. She's like, oh, that's moldy, right? And I'm looking at it and I'm smelling. I'm like, it smells like bread, right? And I'm like, I don't see anything, right? She's like, it's moldy. I'm not eating it. And I'm like, it's a brand new loaf, right? What the hell, right? Looks like I'm eating about 20 sandwiches, right? Crazy, all right? And that is something that has persisted. She still has a pretty good sense of smell that, I guess she had a pretty good sense of smell before that, but it's better than it was before she got pregnant. Any moms out here? Yeah, do you agree? Yeah, you could smell stuff better? For sure. You're like, get out, you stink, right? It's interesting, right? And so that makes me back it up and think from a mom's perspective, if you've got this little bun in the oven, you want to do everything you can to ensure that this little thing stays healthy before it hatches out into the world. And then once it does, you want to keep it healthy as well, right? But while you're still growing this sucker up, it makes sense that your sense of smell might be heightened. So you could tell that maybe this thing is foul. This could be bad news, right? And I don't want to get some sort of crazy infection that could hurt my little growing bun in the oven, right? It kind of makes sense to me that that would be a mechanism to help ensure that you're staying a little healthier, right? Now I'm just like, smell this bread, right? Is it good, right? I don't do that, but you never know, all right? It also maybe depends on the type of bread you are consuming, right? Some bread's got quite a bit of preservatives in there and the potential for it to get moldy. Maybe not, right? Others can get moldy pretty quickly, all right? That's kind of interesting, all right? You've all probably heard stories of people getting some fast food hamburger and maybe leaving it or forgetting about it and seeing it months or years later, and it looks exactly the same, right? That's a little bit disturbing, right? The fact that it's unchanged, still edible, I wouldn't recommend it, but it probably is, right? That makes you wonder, was it ever edible in the first place, right? Kind of crazy, right? Who knows? All right, so shifting over, this is a handful of other types of fungi. Aspergillus in the middle bit is a relatively common one. It can cause some toxic issues, black mold, toxic fungus, sick building syndrome. There's all sorts of potentials where some fungi could be culprits. There's other chemical agents that might serve as some sick building stuff too. But these relatively common, maybe in a poorly ventilated bathroom or something like that, right? Talaromyces flavus, this is another image, and this is just showing some of the reproductive structures with that. We don't need to get bogged down. The third image are some yeast cells. This is Candida albicans, the culprit for yeast infections, candidiasis or thrush. There are some other Candida organisms that can be more of a problem, not that this isn't a problem. This can potentially cause other issues as well, but some other species can be more systemic and get into the circulatory system and cause some severe potential fatal infections. So these yeast cells, and they've got an opportunity to produce a little bit of pseudohyphae. I think we'll see some more of that a little later. It's not a good image there. This looks, to me, seeing this image, looks like some microscopic peas. That's kind of what I think with that, little peas in a pod. These ascospores, the ascus is the pea pod and the ascospores are the peas, if you will, in that pod, all right? Just another type of reproductive structure with those. So this image, another one with the ascomycetes and the way that they are reproducing asexually and sexually, again, forming that union between the opposite mating types as we move through there. The basidiomycetes, these are things that maybe we're a little more familiar with, all right? These are mushrooms, all right? And there are all sorts of different types of mushrooms, things that we potentially may be consuming, things that if you do consume could potentially kill you, right? All sorts. There's a whole spectrum when we think about some of that stuff. And, you know, there are lots of mushrooms that are edible, and you could go foraging. And there are also some that look very similar to the edible ones that are very toxic. So I recommend that if you do that, you truly know what's up, right? And you don't just start picking them and eating them, right? Because that could be the end of you, right? But what if it was a psychedelic one, right? That's also, you know, really kind of interesting to me from that standpoint. There's all sorts of things that are produced as maybe a deterrent to prevent things from maybe consuming it, right? And then you flip it around and you're like, oh, but maybe that has enticed people to eat it, right? They're like, oh, we want to eat those, right? When deep down, you probably would think you should avoid that, right? Who knows, right? all sorts of crazy things, all right? I did research with fungi, but not this type of stuff, all right? It was some of the dimorphic fungus, all right, that gets into your lungs and causes issues. Nothing like this, all right? I think they're interesting, but I'll leave them alone, all right? If I'm out hiking around, I'll leave them alone because I have no clue what they are, all right? You just never know, all right? I've got a friend that was hunting, I think, morels, flipping around logs and rocks and different things and finding, I'm just like, I don't know how you do that, right? How did you know, right? This is whatever, right? But he had all sorts of them. And I was like, wow, that's impressive, right? And right around the corner on the other side of the hill, that's where the psychedelic ones are, right? Who knows? All right. They're all over the place. All right. So this table just breaking it down into some of these. Amanita is a bad one. That's the death cap. And in turn, all sorts of potentials, all right, for this. Puffballs and others that fit into this. Cryptococcus also fits into the Basidiomycota category, but it's a little different and, definitely odd compared to these other fruiting body structures, all right? Kind of impressive. All right, we're getting close to the end of this. So here we've got trichophyton. This is a ringworm infection. So trichophyton rubrum, a dermatophyte for handfuls of different types of fungal infections in the skin. But we can see some of the hyphae in the background sort of blurred out. It's the reproductive structures that we're really looking at. So this macroconidium being big structure and the little individual microconidia released out. Shifting away from the fungi, some dinoflagellates, all sorts of different potentials with these. They've got cellulose armor, some flagella coming out of them, and in turn, giving them aquatic sort of situation, ability to move through. We're looking at an electron micrograph. All of these little bits are the screen, if you will, that this cell has been put on to stain, which is pretty interesting. So all sorts of different potentials when we shift over and think about algae, multicellular things like kelp, and others as we move through. We talked about some potentials with some auger production coming from some of these. Diatoms, I think we showed this image a couple of chapters back, and they in turn have more of a kind of glass-like outside exoskeleton, if you will, that can be used in organic gardening, right? And then some other potentials with them. All right, this Chlamydia monas, this is a protozoal or a green algae, not protozoal, but a flagellated green algae. And I think lastly, I'm pretty sure, this is some lichen. And if you've gone hiking around, you've probably seen all sorts of different types of lichens. Kind of depends on your circumstances as far as where you're at. This one right here, I see this stuff sometimes, I think moss, right? But it's not moss, it's a type of lichen, right? And it's got a little bit of toxins in there that was used to make some like poison darts or arrowheads. Kind of interesting with that. All right, that's the end. Farewell. Yes. Heck yeah, we do. I'm going to talk about some acellular organisms Wednesday and Friday, and then the exam opens up over these 3 chapters. Good?